Development of prefrontal neuronal circuitry in early preterm infants. The early preterm period is important for human development because preterm infants older than 24 post-conceptional week can survive and thus become exposed to interaction with environment. In addition, sensory stimulation is possible even in utero. Structural organization. The prefrontal cortical regions gradually differentiate while transient fetal zones change their appearance and thickness. An initial lamination appears in the middle of the cortical plate, CP. Figure 2.1e. The subplate, or SP, is at its developmental peak and shows regional variations in thickness. The cytoarchitectonic immaturity of the early preterm prefrontal cortex is also visible in the MZ marginal zone, which has several sublayers and contains well-developed but transient subpial granular layer. Transient lamination of the prefrontal cortex in human preterms was well documented by acetylcholinesterase ACHE histochemistry, Kostovich 1990. There is a transient columnar arrangement, figure 2.2d, of the strong acetylcholinesterase staining in the prefrontal cortex with regionally characteristic distribution. The differences between orbitolateral, dorsolateral, and orbitomedial cortex become more obvious and roughly correspond to the incipient gyral landmarks. In summary, structural data on laminar, regional, and radial organization show transient patterns of organization of prefrontal cortex in preterm infant. Neurogenetic events. The proliferative zones, ventricular and subventricular, in preterm infants gradually cease to produce neurons. However, the bulging of the ventricular zone, the so-called ganglionic eminence, remains thick and voluminous. According to the evidence obtained in primates, this zone produces predominantly obtained in primates after 24 post-conceptional week. This zone produces predominantly glial precursors. In our Golgi studies, we have seen many migratory neurons in early preterm infants. Therefore, it is possible that in prefrontal pallium neurogenesis and migration lasts several weeks longer than in primary cortical areas. While there is cessation of proliferative processes, both pyramidal and non-pyramidal neurons continue to differentiate. Subplate SP neurons continue to grow and express different neuropeptides such as neuropeptide Y, MPY, and somatostatin, as well as GABA. The distribution of synapses changes significantly after 24 post-conceptional week. Synapses begin to appear within the deep portion of the cortical plate, CP, and bilaminar pattern of synaptic distribution gradually disappears. The number of synapses in the deep cortex, SP plus deep CP, is higher than in the superficial cortex, superficial CP plus MZ. The predominance of deep synapses in early preterms and deep to superficial synaptogenesis after 28 post-conceptional week are structural factors important for generation of cortical dipole from surface to intermediate zone and changing surface positive potentials. Reference Molliver, 1967. According to our studies, the most intense neurogenetic process in the preterm cerebrum is growth of projection and callosal pathways. Functional organization. The main feature distinguishing preterm cortex from mid-fetal and late fetal cortex, figures 2.1b, 2.1c, 2.1d, is the presence of strong thalamic input from medial dorsal nucleus. In the primary sensory areas, the thalamic input is anatomical basis for evoked potentials. After 24 post-conceptional week, thalamic afferents establish synaptic contacts with the cortical plate neurons. In the somatosensory cortex of preterm infants, pain stimuli from the skin may provoke cortical response as detected by infrared monitoring. That's Fitzgerald 2005. It is not known whether information from primary cortex in early preterms is conveyed further to prefrontal cortical areas. This seems to be unlikely because of the immaturity of cortical pathways. Thus, the prefrontal cortex in early preterms receives predominantly non-sensory information via mediodorsal prefrontal projection. Thalamic axons in early preterm infants make synapses with cells in both subcortical plate and the cortical plate. The contact of thalamocortical axons with subplate neurons is a powerful activator of transient endogenous cortical circuitry. The initial contact with cortical plate cells is a forerunner of extrinsic circuitry. Prolonged coexistence of these two types of circuitry, the extrinsic and the transient intrinsic, is the salient feature of cortical development in humans. The gradual changes in organization of transient and quote-unquote permanent circuitry may form the basic framework for changing EEG. Slow activity transients, SAT, are generated as early as 24 post-conceptional week, transform at 30 post-conceptional week, and disappear after birth. Excitatory extrinsic cholinergic input to the SP subcortical plate also originates in the basal forebrain. Subplate neurons are glutamatergic and GABAergic or GABA-peptidergic neurons. So the references for the subplate neurons are glutamatergic, Antonini and Schatz 1990, and GABAergic, Meinecke and Rakesh 1992, or GABAergic peptidergic neurons, Ellendorfer and Schatz 1994. There is a significant increase in number of peptidergic neurons in preterm infants. While functional relationship between thalamic axon subplate neurons and cortical plate neurons in primary cortical areas was clearly demonstrated in experimental studies in carnivores, Ellendorfer and Schatz 1994, and rodents, 2002, different group. The prefrontal circuitry at this early age was not studied in monkeys, which may serve as a possible model for human circuitry. It would be of particular interest to reveal interaction of quote-unquote extrinsic and quote-unquote intrinsic circuits at synaptic and transmitter levels and correlate this with cortical potentials. In early preterm infants, quote-unquote intrinsic transient circuitry of the subplate may still be the main generator of spontaneous endogenous oscillations and spontaneous activity transients. The transient circuitry of the subplate in the frontal lobe may have output to caudate nucleus and contribute to generation of movements in the preterm infant. It's Goldman Rakesh 1981. One of the most intriguing questions is when, in the human prefrontal cortex, GABAergic neurons become inhibitory. In experimental studies, GABA was shown to act as an excitatory transmitter during the early phases of cortical development. Due to the immaturity of KCC transporter, the switch to excitatory function occurs relatively late in the rat at the age corresponding to neonatal period in humans. I'm not sure if that means a switch to the inhibitory. Let me read that paragraph again. One of the most intriguing questions is when in human prefrontal cortex GABAergic neurons become inhibitory. In experimental studies, GABA was shown to act as excitatory transmitter during the early phases of cortical development due to the immaturity of KCC transporter. The switch to excitatory function occurs relatively late in the rat at the age corresponding to neonatal period in humans. Let me at least look up what KCC is. Okay, so the reference with KCC is slow endogenous activity transients and developmental expression of potassium chloride co-transporter 2 in immature human cortex. So it's potassium chloride co-transporter. Anyway, it says excitatory for the switch. I don't know if they mean inhibitory by accident. Very little is known about connections of prefrontal cortex with basal ganglia and brainstem. In the non-human primates, cortical striatal pathways terminate in full register with striatal cytoarchitectonic compartments at embryonic day 105. Goldman Rakesh 1981. In humans, striatal cell islands and acetylcholinesterase patches appear at 10 post-conception week and become maximally developed between 28 and 32 post-conception week. The efferent pathways from the frontal cortex form the Murattov's fascicle, and this fiber system seems to be the source of afferents to well-developed moduli in the human fetal putamen. Reference for that was Petrides and Pandya, 2007. In the context of transitory cortical circuitry and connections, it is interesting that the first general variable movements appear during the preterm infant period. Hatter's Algora, 2007.